partly, not very far, through packet 2. And we really just started talking about thyroid hormone specifically. And thyroid hormone is a really good hormone to look at in terms of the axis and the function, because the effect of thyroid hormone is really widespread. We talked about it being a hormone that influences metabolic activity of pretty much all of our cells. And we left off, I believe, on this slide, looking at the structure of the thyroid gland and speaking about the follicle. So the follicle we identified as the structural unit of the thyroid, meaning that we have many, many follicles that make up the gland itself. And then we spoke about the cells that line each of these follicles. We called these follicular cells, and these are the cells that are actually responsible for absorbing this colloid and making thyroid hormone. We also mentioned that colloid was nothing but the storage form of this thyroid hormone, which we called thyroglobulin, and that's the precursor that we use to make this thyroid hormone. We also mentioned the importance of iodide. So iodide, which comes in through our diet as iodine, and then it's converted to iodide. That is vital to the production of this thyroid hormone because we use that, combine it with thyroglobulin, and then we make thyroid hormone there. So we cannot make thyroid hormone in the absence of iodide, combining with that inactive thyroglobulin. Okay, so that's where we kind of left off, and we're going to pick up from there, look more closely at the cellular process that's involved in making this thyroid hormone, and then we'll talk a little bit more broadly about the actual thyroid hormone axis. Okay, and then as usual, feel free to stop me if you have questions. I'll be monitoring the chat. Okay, so this is a closer look at one of those follicles. So this is an individual follicle. This is a follicular cell that's lining that follicle. And here we can see on either side of the follicle, we've got the colloid, which is inside the follicle, and then we've got the blood vessel, which is around the periphery of these follicles that's running through this thyroid gland. So the big picture here is that we want to absorb this inactive form of thyroglobulin. We're going to make thyroid hormone within the cell, and then we're going to exocytose this thyroid hormone into the bloodstream, and then that can be diffused throughout the body and go to its individual target cells, which happen to be all of our cells, and then influence metabolic activity that way. So we start off with these residues. So we've got diiodotyrosine and then monoiodotyrosine, which is this DIT and MIT residues that basically latch onto this thyroglobulin and form thyroid hormone. If we've got 3 of those residues, it's T3. If we've got 4 of those residues, it becomes T4. So we have these 2 different versions of thyroid hormone that are formed here by combining this iodide to the thyroglobulin to make either T3 or T4. Now that T3 or T4, I'm getting tongue-tied, is then brought into the cell, the follicular cell, by endocytosis. It is combined into this vesicle, and then it moves across the cell until it gets to the apical surface of this follicular cell. And here we can see that it moves freely across the plasma membrane. Now that tells us one thing, that this is a lipid-soluble or lipophilic hormone. It does not require any channels or transporters or carriers to move across the cell membrane. And it can go right into the bloodstream and then be diffused throughout the body. The second thing that we identify here is that this enters the bloodstream as either T3 or T4. Again, depending on how many of those residues were combined initially. And then in the bloodstream, T3 is the active or bioavailable or bioactive form of this hormone that is easily absorbed into cells and used, whereas T4 is more of the signaling version of this hormone. And I'll explain a little bit later what that means. But T4 can be converted to T3, and T3 is really the magic. T3 is really the bioavailable form of the hormone that can be readily used by our cells. The second thing I want to point out here is the way that this happens, the stimulus for this process. This is TSH. So TSH is thyroid stimulating hormone. This comes from the anterior pituitary. This is one of our tropic hormones. And this binds to this receptor, which we should already be identifying this as a metabotropic receptor, being that it is coupled to a G protein. It turns on this amplifier enzyme downstream, and that actually generates some second messenger. In this instance, it's cyclic AMP that can turn on protein kinase, and that is what stimulates the process of bringing in this T3 and T4. So the stimulus for thyroid hormone production is TSH, which comes from the anterior pituitary. And that's what we mean by saying that TSH is a tropic hormone. It is going to stimulate the release of another hormone via this, this mechanism that we just described. So that is how we use TSH from the anterior pituitary, stimulate the thyroid gland to then release T3, T4. T3, T4 will enter the bloodstream, being that it's a hormone, and it will flood the bloodstream and have its various response. Okay, we'll talk about some of the tissues that it actually targets. Okay, so just to summarize here, again, the actions of thyroid hormone, it is lipophilic, so it's going to behave like a steroid. It's going to be fat soluble and move through our membranes really easily. It's going to alter the rate of transcription of our mRNA. And when we say the rate of transcription, basically the metabolic rate of that cell. If we start making mRNA faster, the cell is going to speed up its activity, speed up its metabolic function, and that is the function of thyroid hormone. Now, an important clinical correlation here is what we call cretinism, and this is a disorder that comes about if there's insufficient exposure during fetal development to thyroid hormone. So cretinism we see usually in infants that are born and the mother didn't have enough iodine in her diet. And so we see an insufficiency of thyroid hormone during the fetal period. And so this ends up presenting with cognitive disabilities, learning disabilities, sometimes facial abnormalities, and that's all due to the absence of thyroid hormone in sufficient quantities during fetal development. Okay, all right. Okay, so here's a nice list of the actual functions of thyroid hormone. So it has this calorigenic effect. And I was sort of talking about that during our last session where we can see that exhibited in patients that have hyperthyroidism. They have really fast heart rates, lots of sweating, increase sometimes tremors of the hands, increased metabolic activity, increased skeletal muscle activity, weight loss we also see, and that is indicating the calorigenic effect of this thyroid hormone. So it does a lot of things in the cell that speed up the metabolic rate of the cell. Things like increase the rate of the sodium-potassium pumps. It increases the number and size of the mitochondria, making more energy available faster. It increases the rate of oxidative phosphorylation and the enzymes that are involved in oxidative phosphorylation. And so we should be tying that to more ATP production, which means more energy expenditure. It enhances processes like glycogenolysis, which is the breaking down of glycogen, proteolysis, the breaking down of proteins, and lipolysis, the breaking down of fats, which again is liberating more energy to do this cellular work at a faster rate. It's also going to promote gluconeogenesis, which is converting proteins or amino acids into glucose. Again, another mechanism of liberating energy, and then ketone synthesis as well. And then it promotes synthesis of beta-adrenergic receptors, and that is going to also help with speeding up the metabolic activity. We know that beta-adrenergic receptors are associated with the sympathetic pathway and the sympathetic influence on cells, which is speeding up the heart rate, speeding up the metabolic activity. So all of these different cellular influences really speak to speeding up the rate of energy production, speeding up the rate of energy usage, and then speeding up the breakdown of those biomolecules to liberate energy. okay? And that is what thyroid hormone does. And that's why we don't want to have it in excess, but we also don't want to have it in insufficient quantities. We want to have a nice balanced amount of thyroid hormone so that we can keep our metabolic activity at a steady rate. Okay, so let's look at the axis that I spoke about. So this is just an example of one hormone and its axis. But we can sort of apply this general principle to, to all of the endocrine hormones. So we're gonna look at the thyroid hormone axis. And how the different glands and hormones bring about this thyroid hormone function. So we start up here with the hypothalamus. Okay. And then we've got the actual thyroid gland. So this is what our thyroid gland looks like. We've got these two large lobes with 4 poles, right? This is our thyroid gland. And then lastly, we can speak about some of the target tissues as the final structure involved. One of the major ones is going to be the liver, and then the kidneys as well, but we'll just draw the liver here for simplicity. Alrighty. So we know that the first part of this is the fact that the anterior pituitary secretes a thyroid stimulating hormone. We sometimes call it TSH. So the hypothalamus, oh, we forgot to label the pituitary, sorry. So the anterior pituitary is going to be this distal part of this axis here. Okay. So the hypothalamus influences the anterior pituitary via the release of TRH, so thyroid releasing hormone, and I'll abbreviate that as TRH, coming from the hypothalamus, stimulates the anterior pituitary to release its hormone. So this is going to be a positive stimulus here. And the hormone that's released from the anterior pituitary is called TSH. So TRH is thyroid releasing hormone coming from the hypothalamus, and then TSH is thyroid stimulating hormone coming from the anterior pituitary. Now that is also going to trigger the release of thyroid hormone. Let's make this black, which is signaled from that TSH. And we kind of looked at that specific example with the metabotropic receptor and how TSH stimulates the gland to actually make thyroid hormone, okay? So that's the second part of the axis here. The third part is that the gland is going to secrete its hormones, which we described as being either T3 or T4. So this is T3 or T4. Being secreted from the gland. And that T3 or T4 I described as being a little bit different in its function. So T3 is going to be the more bioavailable form that goes to our target tissues. And I'll put 2 plus signs here because it's going to be really the main active form of this hormone on tissues like the liver and kidney, whereas T4, when it enters the bloodstream, it will then be converted to T3. So T4 in the bloodstream is converted to T3. Okay. T4 is also involved, I'll put here, also involved in the negative feedback. Okay, so T3 is really absorbed by tissues, and these are going to be things like the liver, so the kidneys, liver, and then peripheral cells. All of those functions that we talked about, such as increasing mitochondrial production, increasing oxidative phosphorylation enzymes. So these are the things that T3 is doing in each of these tissues. So our peripheral cells, increasing the metabolic production of our liver to break down those biomolecules and liberate energy. Okay. Now let's talk about the feedback axis of this. We talked about this briefly during our last unit, where we have this long loop negative feedback and then a short loop negative feedback. So the short loop negative feedback is TSH feeding back, let's make this red, feeding back onto the hypothalamus here and telling the hypothalamus to stop its secretions. It's going to be a negative signal. And we describe this as being a short loop because it's coming just from the anterior pituitary onto the hypothalamus. Okay, so this is our short loop negative feedback. And so if we have excess quantities of TSH, then we can slow down the production of TRH. And so we turn off that production via the short loop negative feedback. And then we also described a long loop negative feedback, which is basically coming from the actual third hormone here. So from the thyroid gland, T4 specifically, so I'm going to circle T4. T4 specifically is what is doing the negative feedback here. And it is going to go back again to the hypothalamus and influence the hypothalamus to turn off its production of TRH. And so we describe this one as being a long loop negative feedback. Oops. And in brackets, I'm going to put T4 here because that's really the form of this hormone that is involved in the negative feedback. Okay, so this is how that axis that I spoke about, let's put this closer here. This is how that axis that I spoke about is really controlling or regulating the production of these hormones. So we don't want this hormone in excess and we don't want it to be in insufficient quantities. And so the feedback mechanism here is really keeping a tight regulation of how much hormone is present in the blood. And that's why when we have cases like hypothyroidism or hyperthyroidism, we see excess quantities of sometimes T3, sometimes T4, sometimes TSH, or insufficient quantities of these hormones because of certain pathologies that bring these states about, okay? But in normal, healthy physiology, we have a tight regulation of these thyroid hormones and then the tropic hormones that are involved.